So my name is Ben, and I'm presenting what will be a chapter from my dissertation today about the northern red belly and fine-scale dace. It's a bit of a boring title, but I like to think it's some cool fish that do some neat things, so I'll try and give you a succinct summary of what's going on with this system. So the two fish, Chrysomus eos and Chrysomus neogaius, the northern red belly and fine-scale dace, are fairly common leuciscid minnows that you can find across a lot of Canada. And its range kind of encompasses northern North America. And so they're pretty beautiful fish. They have some nice coloration during breeding season, but they do something very remarkable, and that is that they hybridize and form this really interesting unisexual F1 hybrid. So all of these are female, and what makes them more remarkable is they're one of only about 100 species that have been identified that are vertebrates that can reproduce asexually. So they actually reproduce using a really complex form of, I think, parthenogenesis called gynogenesis. And so what that does is they produce a diploid ova that actually requires stimulation by the sperm to continue to progress, but it doesn't actually incorporate the material from that sperm, very often at least. What makes it more complex is sometimes they do return to syngamy and they produce these triploid back crosses, which then go on further and produce diploid-triploid mosaics. And finally, we end up with these interesting hybrids where they have the mitochondrial genome of the mother species, but then the nuclear genome of the paternal species. So, a bit of background about what's been discovered so far. It's been estimated that these fish hybridized for the first time during the last glacial maximum of the late Wisconsin era. It's put at a max of about 50,000 years ago, but at some point in a glacial refugia in the Mississippi Valley, they hybridized, forming this asexual lineage, or multiple asexual lineages. And then as that glacier receded, they penetrated out and recolonized the new freshwater landscape alongside the parental species. So these F1s might be these relic populations from this ancient hybridization. And earlier papers suggested that there's this extreme uniformity amongst these F1 lineages, which would suggest that they're all related to this ancient hybridization. But more recently, it's been determined that there's actually evidence of these hybrids being formed in situ, so occurring alongside the parents in the geography they're in today, as well as some evidence of contemporary formation of these asexual lineages that's been put forth due to climate change. So what I wanted to do was take a new approach using a reduced representation sequencing approach, and I looked to answer, are all these F1 hybrids really just these asexual females of clonal origin that are being produced or maintained from an ancient hybridization event? Is there any evidence, for example, of historical backcrossing, where maybe these two parental species have hybridized and formed sexual hybrid offspring that have then backcrossed back into the parental species populations? Or is it potential that there are sexual hybrids occurring alongside the asexual hybrids as well over time, including being formed today? So to do this, I started by sampling across Ontario. So I had 6 sites in Ontario, 2 in Algonquin Park, 1 in Sudbury, and then 3 others in rural southern Ontario. And then collaborators in Wyoming actually sent us some fine-scale dace from a couple sites in Wyoming. When I captured these fish, I phenotyped them based on morphology, and then I also sexed them based on strong determining sex characteristics. The beginning of my lab work pipeline, I took fin clips and I extracted DNA using QIAGEN D&EZ blood and tissue kits. And then I prepared GBS libraries and sent it off for sequencing at TCAG at SickKids in Toronto. And as a result of all my alignment to our northern red belly genome and filtering, I ended up with 308 individuals and 41,862 SNPs. So further, I did ploidy analysis because we know there's some really weird things occurring with ploidy. And I used flow cytometry on the second pectoral fin. So the first one I used for the DNA extraction, the second one I used for flow cytometry. And what was really interesting was using this methodology, I didn't actually identify anything other than diploids. Anything that was slightly above what I would expect the genome to be sized for a diploid. I went further and did full-gen image analysis densitometry, and even that determined they were still diploids. So I didn't actually find any of those rarer ploidy offspring. My bioinformatics pipeline started using a program called Entropy. And so on the bottom of the x-axis here, it's a Bayesian hierarchical mixed model that estimates two parameters. And so on the x-axis there, the first parameter it estimates is the admixture proportion, or Q, which is roughly analogous to what you would get from structure or admixture. And that gives you the proportion of ancestry from either parental species that you're looking at. So if you look at the 0, that would be parent species 1, the 1 is parent species 2, and then the 0.5 would be a hybrid, and everything in between is backcrossing or other admixture. And then on the y-axis is the big Q, aptly named, and that is the intersource ancestry complement. And so you can imagine that when you have an F1 hybrid, at each loci you have two alleles. And that F1 hybrid has one chromosome from either parent species, so you would get a value of 1 because each of those alleles comes from one parent species. The really great thing about the big Q, though, is it allows you to discern between generations of those hybrids. So you can see F1 hybrids, F2 hybrids, F3 hybrids, and beyond. Whereas if you only have that Q score, you'd only see that flat axis, and they'd come out looking roughly like F1 hybrids. So the first bit of results to share, what is that admixture proportion, that little q value for all of my fish. So this is all 308 fish that have been sequenced and then put through entropy. And what you can see there is quite a gradient of admixture. I've boxed here what are roughly estimated to be probable F1s or potential F2s or F3s. But what I might have expected was more of a plateau there if it was just these asexual F1s, and then rarely you'd get maybe a little bit of that admixture with those strange ploidy back crosses. Going further, these are the triangle plots that show both the little q and big Q values, and I've broken it out by location here. So I only have 7 locations because I combined the 2 locations in Wyoming into one. And what you can see here, it might be tough to make out, but so there are F1 hybrids at the top of the triangles, as I expected to see. But then there is also some F2 hybrids and then some really interesting individuals where the Q score is near 0 and they lie at the bottom of the y-axis, but they're at the 0.5 of the x-axis. And then we have a fair amount of backcrossing here as well. And again, with the Wyoming population, what's really interesting there is there were no northern red belly there, or there shouldn't be, at least there weren't any captured and identified. So that means that it could be those hybrids that have persisted alongside the parents creating that admixture. Okay. So the next program I used was Bayesian genomic lines, and I used this to estimate patterns of locus-specific introgression amongst the genome of the admixed individuals that I have. And so the figure that you're seeing on the right here is the mean alpha values for 2 populations. On the lower right, you're seeing Lake Eos in Sudbury, and on the upper left, you're seeing, or Lake Eos in Algonquin, and on the upper left, you're seeing Silver Lake in Sudbury. And so alpha is the Klein center, and a positive or increasing value or negative or decreasing value of alpha represents that probability of ancestry at that locus, and then beta is the Klein slope. And again, the positive or negative is corresponding to increasing or decreasing rates of transition between the probability of ancestry. So here, what you're looking at is differences in that mean alpha value, and I've done the Pearson correlation that shows very little correlation between the two, suggesting that at these 2 locations, the lineages are separate. They aren't related to one common hybridization event. And that's further exemplified by these, where I've plotted them both alpha and beta on these topographic graphs, and what you're seeing there is quite a bit of differentiation in how those loci are coming out on these charts, suggesting again that these are hybrids, admixed individuals that are not related to each other, and they've likely been formed in situ. And then finally, the last thing I did was taking the sex results that I have, so the individuals that I phenotype based on sex, I ran those as my admixed individuals in BGC. And what you're seeing on the left and right are these outlier Kleins. So if you look in the middle, that red line represents kind of a null expectation. If you have alpha and beta set to 0, then that red line is equal to the proportion of ancestry, that Q value. So on the bottom, you have that hybrid index or proportional ancestry, and on the y-axis, you have the probability of ancestry at those loci. And so being outlier loci, these are loci that are outside of the 0.995 or 0.005 quantiles. And plotting them here, what you're seeing on the left are males, and the alpha and beta values for those outlier loci are very narrowly going away from that kind of null expectation of an F1 hybrid, meaning that the male admixed individuals I had that came out as F1 hybrids are likely contemporary and were formed by hybridization events occurring between the two parental species in the location I sampled them. Whereas on the right, the females are coming out really kind of wacky, and the loci are doing some really strange things. So to try and describe it, if you look at these huge sloping lines, right, it's suggesting that even at incredibly low ancestry, you have 100% probability of inheritance of that loci from the northern red belly days. So on the right of the x-axis is the northern red belly dace as a species. On the left of the x-axis is the fine scale dace as a species. And then that one marker would mean 100% of those loci that are coming out have ancestry from the northern red belly dace, which is fascinating. And that might be because those females include some of those asexuals that I would have captured. So some conclusions, I found hybrid individuals at all of my sample sites, and a diversity of admixture and intrasource ancestry amongst them, including a lot of backcrossing. The admixed populations did not share a common origin, which confirms what has been found, that they aren't just these extremely uniform clonal hybrids. And then I did find contemporary male F1 hybrids, which I think is the first time they've been identified amongst these populations. And so to acknowledge all the people listed there in my lab and collaborators, as well as my wife and son and the funding sources I had. That's it. Thank you. Might have gone a little fast there, sorry. Got 3 minutes for questions, if anyone has any. Yeah, like the bottom right one. I'm not certain. I guess my assumption would be that some of those hybrids coexisted alongside just the fine scale dace and must be backcrossing into the population. It seems to be fairly recent, so I don't know if they're not catching any northern red belly. I don't know how they would hybridize and then backcross. I've, coming up with reasons. I could speculate that maybe bait fish, like if someone's dropping a bait bucket, for some reason they have northern red belly, but I really am not entirely certain what might be going on there, aside from the presence of these already hybrids that follow them over there. If that makes sense. Any other questions? Did that kind of get at what you were asking, I guess? Yeah, I'm unsure of why. Like I said, like they're fairly certain, the people that collected these for us, that northern red belly don't exist there. So it's a bit of a conundrum as to why they are there, but at least we know there is some mechanism of these hybrids following the parents. So, yeah. Okay. Well, if there's no other questions, then
I'm going to be sharing some work that we've been doing on hybridization in a different group of minnows from what Ben just talked about. And so before we go farther, I want to acknowledge that this is not primarily, well, I'm not the one who did the bulk of the labor here. This is a master's thesis from my former student Amanda, who's now moved on to do a PhD in Stockholm University, so she couldn't be here. And with collaboration from Amy Patera, who is here today, so go talk to Amy about these fish. She's working on different questions in the same species. All right, so we have this extraordinary biodiversity of fish worldwide, and it's maintained by evolutionary processes. And so I've put this picture here because it shows that when fish spawn, they're not always like very carefully choosing. They share nests. There's all sorts of beautiful breeding behavior in leuciscid minnows. And so the question is, how are species maintained separately from one another? And so this brings us to this whole, this is a hybridization session, right? Everybody here is interested in this. But when we have species that are diverged from one another and they come together, breed in the same place, there's some possible outcomes. And so there could be that reproductive isolation is completely maintained. This has to happen at least a lot of the time to maintain our divergent species. But we also, a lot of the time, have a scenario where hybridization occurs, or potentially the collapse of parental species, or even hybrid speciation in some special cases. And so what I'm talking about here fits firmly in that contemporary hybridization category. And so one thing we want to think about is how consistent hybridization is across locations. So this is a river network, not a phylogenetic tree, for the record. So you could have a scenario where hybridization happens kind of the same everywhere a pair of species comes into contact. If you've got something like an innate genetic barrier that is driving your outcomes, that's kind of what you'd expect. But in reality, we often see that we can have variation in outcomes of hybridization. Either some places they hybridize, some places they don't, or some places they hybridize a lot, like F1s, F2s, back crosses, like the whole blur of ancestry between species. And in some places, it's more constrained. And so we've seen in a lot of systems now that hybridization does tend to vary across replicate locations in a lot of fish. The particular example that I'm talking about today is hybridization among leuciscid minnows, specifically hybridization among leuciscid minnows in southern Ontario. And we're interested in this area specifically because a lot of the habitat looks like that lower left picture. These are lovely, pristine habitats that are not at all mangled by agriculture and urbanization. Just kidding. These are highly disturbed areas, and we have this expectation that disturbance could shape hybridization either by increasing opportunities for hybridization, such as if there are fewer suitable spawning sites, or if it somehow changes the fitness landscape of who does well and makes it so that hybrids are more likely to be successful. And so that's kind of the conceptual backdrop of this work. Another thing that's specific to this group of fish is that they have a variety of breeding behaviors. So here's another picture of a whole bunch of different species all spawning together. There are also some non-leuciscids. But we have a scenario where not all of them build their own nests, and so they use the nests of other related species, which is kind of a prime opportunity for hybridization. So we're interested in this case how much hybridization is happening. There was a recent study that suggested that breeding behavior drives which species hybridize and which ones don't. And so that's kind of our backdrop as well. So we've got this system where we've got potential hybridization between a bunch of species in a disturbed environment with a whole bunch of breeding behavior. So we targeted 9 different species, sampled from 29 locations. These locations span a range of anthropogenic disturbance, but it's important to note that most of the areas we sampled are upwards of 60% agricultural. So it's very hard in these areas to find anywhere that's not agricultural or urbanized. We have comparisons from Algonquin Provincial Park, which, to our eyes looks a bit more pristine, but was indeed heavily logged about a century ago. And so truly undisturbed locations are hard to come by. We generated genotyping by sequencing data for these fish. And as part of this work, we've also created a novel reference genome for the creek chub. This is a whole separate study that I won't really get into, but if you work on these fish, this is out there, you can use it. Papers published. So it's a nice genome. It came out well. And so we aligned to that genome, and then we completed analyses of hybridization using entropy, which Ben has already talked about, so I won't go into the details. We found actually that although we had targeted 9 species, we ended up with k equals 12 because there were previously unidentified species that had gotten into the act as well. Specifically, we had some Pomephilus species that were involved too. And so what was really striking about this outcome is that 29% of our individuals were hybrids in this sampling. This seems really high, but we don't have a point of reference for if this is what typically happens. When we look at the distribution of these species and their hybrids across sites, so the takeaway here is not that you'll be able to see individual colors and track your favorite fish. The takeaway here is that we have different suites of minnows interacting in different places and producing different hybrids. Another takeaway here is that there are 2 species hybrids So these plots are oriented a little bit differently than Ben's, where individuals are horizontal. So a horizontal bar that's half one color, half the other, is an F1 hybrid. So we have 2 species hybrids. They range from F1s to back crosses. There's some F2s in there as well. So there are also some multi-species hybrids, so hybrids with ancestry from usually 3 species. Beyond that, it gets a little bit tricky to identify, and we get a little bit more suspicious about our models, but there are definitely some 3-species hybrids in there. We also found some previously unknown hybrids, and we found the phenotypes quite cryptic, actually. So a lot of these hybrids were not identified as hybrids in the field. I've put some images of these fish here. Almost all of these fish are about this big, have a black stripe on their side, vaguely the same shape. These are hard fish to tell apart, and so it was known that these could hybridize, but it seems like hybridization is more common than we thought because the phenotypes are difficult. These are not like obviously intermediate fish. But yeah, it suggests that, you know, with a genomic view of this system, we're seeing more hybridization than was previously quantified. We also saw that all sampled species hybridized with at least one other species. And so it is clear that in this group, reproductive boundaries are fluid, and we're getting a lot of hybridization between very different clades. We are interested in incorporating environment as well. And so we collected publicly available data on a whole bunch of environmental variables that we thought could be important for either predicting spawning success or fitness of the offspring. Things like land use, there's a whole wealth of publicly available data for our sites. So we pulled those, used those as predictor variables in logistic regressions. And these are the significant relationships. But what I want to say about this is that it's tricky because we don't put much stock in these, although they're statistically significant. We don't put much stock in these from a biological perspective because of the distribution of variation in the environmental variables. So a lot of these relationships are leveraged by a few points, and so we're not really confident that this is, you know, very satisfying to us. I don't think that we've despaired of figuring out how environment's being important, but it's becoming increasingly clear to us that we need to do it differently. And so things that we've been thinking about are what's the best response variable to use? Like how should we be quantifying hybridization as our response variable? Here we've done it as proportion hybrid individuals at a site. That's nice, but it's very broad brush. It would also, getting the environmental variable scale right is also tricky because you want to have the local conditions, but the local conditions where you catch the fish on the day you catch it might not matter as much as the conditions where those fish are spawning. And so to do a really good job of this, you would need to do seasonal resolution. It's a lot of work, quite honestly, to do a good job of using environment to predict hybridization outcomes. So, I mean, that's one of my long-term goals, but hard work. Another thing we found was that we didn't have a clear connection to breeding behavior categories. So I talked about how these fish have all sorts of cool breeding behaviors, but actually almost all of the parental species were non-garter brood hider, and so we didn't have that resolution to look at contrasting breeding behaviors in these fish and how that predicts hybridization outcomes. Seems like they've got no problem hybridizing regardless of their breeding behaviors here, though. So let's see, to wrap up, this study showed, this is I believe the second genomic study of hybridization among leuciscid minnows. Someone else did a really cool complementary study in Arkansas around about the same time we were working on this. So we've shown extensive hybridization among these minnows in southern Ontario. One thing we'd really like to get at in the future, and something that's kind of on our bucket list, is whether this reflects increased contemporary hybridization due to anthropogenic disturbance, or if there's been historical bouts of introgression. And so we're going to try and get at some of that historical introgression piece next. And in general, we'd love to do some more work on mechanism in this system. There's a lot to do. There's a lot to understand. It's complicated and messy, but I kind of love it. Okay, and so I think I'm going to end there. I'd like to acknowledge our funding sources at the University of Guelph. I'm currently at Northern Michigan, but this work is all in collaboration with University of Guelph students, funded by various Canadian sources and with computing from the Digital Research Alliance of Canada. I'd also like to thank everybody who helped us with this work, and a shout out to the fish. I am currently at Northern Michigan University and will be recruiting master's students over the next few years. So if you want to work on more fish hybridization, maybe in Michigan, get in touch. Okay, and I'd be happy to take any questions. Great. Yep.
Do you have any, this is an agricultural system, right? So
Yeah.
you have the potential to have a lot of different types of pollution running off into these waterways. Do you
Yes.
have any situations where you get the lack of pre-zygotic barriers with mate selection, like not recognizing the same similar mates, so where that happens in like Clemens Lake or those suckers?
Yeah, so you're asking, so this is an agricultural system, lots of like runoff pollution. Is there a problem with them recognizing each other? That could totally, that could totally be a thing. We tried to quantify turbidity because, you know, that's kind of a classic story, like the suckers you referenced, but also cichlid fish, like mate selection gets messed up if they can't see each other, which is not surprising. The answer is that we don't have a clear relationship, and also we weren't necessarily sampling these fish when they were breeding, and so we don't have that, but it totally could be related to mate selection. But they kind of all spawn in a gaggle too, so I don't know exactly how much selection is going on. Yeah. Yep.
So I was trying to think of like these nest associates that
Yeah.
spawn over the nests of these other guys who are making nests. I was sitting there trying to think about what the predictions would be like. Should it be that the guys who are making the nests have mechanisms that are really good at preventing thorn sperm, or should it be the nest associates that are really good at the sperm and egg, matching? I guess
Yeah.
what are your thoughts on that? If you had to
Yeah.
prediction as to who would hybridize more?
Oh, who would hybridize more, the ones, the nest associates or the? Yeah, I would think that maybe, this is pure speculation, but I've wondered about this. I would think that the species that don't build their nests would tend to hybridize more because they have this obligatory use of other species' nests. And I'm out of time, so I'm gonna step down, but yeah, really interesting question.
Hey, so hello everyone, I'm Sofia, and I'm going to talk about hybridization in Iberian chubs, which I realize have nothing to do with American chubs, at least morphologically, although we also have minnows and they're also not very similar. And this is part of the work I've been doing for my PhD at the University of Lisbon. And so I think we can all agree that in the last couple of decades we've come to realize hybridization is way more prevalent in nature than we previously thought, and its role in promoting speciation and adaptation has been demonstrated for a variety of systems. But I think we can all agree that there's still a lot we don't know, and this is why there's a whole day of hybridization stuff. So what I'd like to do today really is convince you that I have a really great system to study it. And those are the Iberian chubs, which, poor fish, they look sort of bent. So the Iberian chubs live in Iberia, as the name sort of indicates, and this is a depiction of their distributions. Most of the species are here. So Iberia, in the western tip of Europe, sort of promotes this really cool gradient of increasing temperature and frequency of drought in these fish distributions. So there's a bunch of species here. I promise you don't have to memorize the names. But what's really cool is that as you travel along Iberia from north to south, annual temperature is higher and frequency of drought in the summer is also increased. So on the left side of the panel, on top what you see is a classic northern river, so in the distribution of the pink species up north, and you see there's shade, the water is flowing. This is in September. I took these pictures sort of a week apart. So it looks like a great place to be a fish. And in the bottom picture, what you see is a classic river on the distribution of the blue triangle species. So it's pretty dry. What you see actually on the left, on the right bottom of the picture is dry riverbed. So the river sort of dries in these separated ponds during the summer, and we all pray and hope that it's going to rain in the winter and it's going to be connected again. And that's sort of the system where they live in the south. And I told you, you don't have to memorize species names, but you sort of have to memorize these four up here, but we can do it with colors and shapes. So northern species is a pink square, and I realize that's not super pink on the image. Then in the middle of the peninsula, you have a blue, light blue circle. Then there's this species with a very small distribution, sort of in the western tip, which is a purple diamond. And then you have a broader distributed species, which is a blue, dark blue triangle. And so these are the ones you really need to care about. And so to detect hybridization in these species, what we have done is we went along and we sampled this fish all over Iberia. And this was only possible due to our great collaborators also in Spain. And we have this sampling scheme where we prioritize having more sampling locations, so covering more of the distribution over having more fish per sampling location. So we sampled usually 2 at least in every location, but yeah, that's pretty much the scheme. And we sequenced their genomes at low coverage whole genome sequencing. And so I'm going to show you first a bunch of admixture plots, which you can see there on the left. So the species are ordered from north to south, so you can follow along in the map that I've put next to it. And just to reassure ourselves, the clusters make sense. They're mostly species or geographical subdivisions within species. So like, for example, for the dark blues in the bottom, it's different river basins that they occupy. So that's all very neat and tidy, and we like it. But I would like to draw your attention particularly to 2 things. One is that when you reach a certain K value, there's this cluster that pops up within the light blue species, the light blue circle species. And this is a population called the Zizr population. This name you sort of have to memorize a bit for the next slides. And you could probably tell me that this is because I sampled way more individuals from this population, which you can see from the left side of the admixture plots. And I can assure you that if you downsample this to 2 fish, you'll still get the same pattern. So they're quite distinct. And then I would also like to draw your attention to these guys over there. They're a special population called San Martinho, and this is part of the purple diamond distribution, so these guys should be all purple in that admixture plot. And I think we can see that they're not all purple, so they seem to be a mixture of purple and blue. Now, if we look at the relationships between the species, this is a nuclear tree, and the really important thing you need to get from this tree is where the blue circle is. So the blue circle is right next to the pink square. However, if you look at a whole mitochondrial tree, and this is the work of Catarina Bernardo, a great master student I work with, you will see that the blue circle is no longer next to the pink square. It's now next to the blue triangle. Another thing is, if we come back to those 2 special populations I told you about, so the Zizr and San Martinho, Zizr was a blue circle population, so they should be next to the blue circles. And that's true for half of them. The other half have this special mitochondria that sort of pops up next to the pink square. And for the San Martinho individuals, what we see is that they were basically purple and blue. So what you can see in the mitochondrial tree is that there's both mitochondria in the population. So there's a purple mitochondria and a light blue mitochondria. So to sort of summarize, because I know this is a lot, before we go into testing for hybridization, what we've seen so far is that for the light blue circle species, there's a mitonuclear discordance, so they group with one species in the nuclear genome and with another species on the mitochondrial genome. Then for the San Martinho population, you have basically evidence of admixture and 2 mitochondria in the population. And for the Zizr individuals, you have 2 mitochondria in the population. So if we start actually testing for hybridization, I'm going to show you a bunch of plots like this. These are D-stats, and you'll see on the x-axis various populations. And on the y-axis, you'll see the d-value. And if you're not familiar with this statistic, basically if your d is 0, that means this is your correct topology, no evidence for hybridization. However, if your d is not 0, then that's evidence for hybridization. And we'll test a bunch of topologies that I'm going to show you on the left of the plot or on the right of the plot. So for the light blue species, starting with this topology, Actually, this is my D value, so it's pretty negative, pretty strong, indicating pretty strongly significant, indicating introgression from the pink square species into the light blues. But you can also tell me this is your mitochondrial tree, this is wrong. What if you test a different tree? So if we flip the tree and then put the blue circle next to the pink square, what you'll get is a still negative, still significant D value indicating introgression, in this case, from the dark blue triangle into the light blue circle. So we think this is evidence for ancient hybridization that has to predate the current species distribution, because the populations I'm showing you here are not actually connected anymore. So they are in different river basins, and the fish cannot move between them. And this is pretty consistent with what we inferred with doing demographic modeling on GBS data, where we inferred that actually the light blue species, about 80 to 90% of their genomes actually comes from the pink square species. Now, if we move to the San Martino population, again, starting with the topology where we put them next to the purples, because they're part of the purple distribution, you'll get a pretty negative D value. And if you flip the tree, you get a still more negative D value, in this case then indicating introgression also from the purple to the San Martino population, not only from the blues. But in San Martino, we were kind of predicting that something was going on in there, so we actually sampled more individuals. And if you look at the result per individual, so individuals are ordered from 1 to 9 on the x-axis, you see that there's quite a lot of individual variation. And we interpret this as evidence for a pretty recent hybridization. We do not find this signal anywhere else in nearby populations, so this is pretty localized. Finally, what about the zither, guys? Well, here, doesn't matter what we try, doesn't matter what topology we test, we find no signal of introgression at the nuclear level. So despite the fact that there's two very divergent mitochondria floating around in the population, we haven't found any signal at the nuclear level. So now that we know we have at least two hybridization cases, one pre-ancient, one pre-recent, we would like to characterize them. And to do that, what we did is we went sampling again, this time a bit more individuals per sampling location at a higher coverage so that we could confidently call genotypes. And what we would like to do here is use demographic modeling to characterize these hybridization cases. So starting with this one, and this is the only one I'm going to talk about. If you remember these light blue circle guys, they had evidence for introgression from both the blue triangle and the pink square. And so really what we wonder is, do they diverge from the pink species? Do they diverge from the dark blue triangle species? And if so, when did the secondary contact happen? How much proportion of each paternal species are we talking about in here? Or could it be an even more weird case where two lineages hybridize and then the hybrids just maintain reproductive isolation from their parents? We don't know because we haven't done the modeling yet. But what I can tell you a little bit about is population sizes over time. So in this plot, you'll see population size on the y-axis and time on the x-axis. And the more you go to the right of the plot, the more back in time you are. And you can see that the pink species and the blue triangles had pretty big population sizes, then they crashed, and then there's a slight recovery. And the crash is more noticeable for the pink species, and then they recovered later as well. And if we think that most of the genome of the light blue circle actually comes from the pink, we would expect that they follow the pink trend, right? Well, actually they don't, so they follow mostly the blue trend. The blue species, and so this actually leaves us with more questions than anything, but we'll work on the modeling and hopefully we'll have some answers soon. And so to summarize, I really hope I've been able to convince you that this is a really cool system to study hybridization because you have multiple cases, some are ancient, some are more recent, plus they're along this really cool environmental gradient with lots of variables we can test. And if you kind of remember the map, the top guys are actually in the middle of the cline, which I think is super cool. And so I think there's really a lot of potential in here. And so finally, I'd like to thank all my lab mates in Vitor Souza's group, my supervisors, Vitor, Felina, and Carla, all the people that helped and the funding sources, and like the fish, of course, yeah. I guess I have time for questions. And no light. Go ahead, sorry.
I have a question too. So sometimes you have flooding events.
Yes.
Could that also be something that's in the history of the species, and that could have been like hybridization events that happened?
Yes, I don't think I can show you, but what I didn't show because I really thought I wouldn't have time and I clearly did, is that Iberian rivers didn't look like this 2 million years ago. So the fish are pretty ancient and ancient compared to the current river drainages. So actually the Iberian rivers used to be lakes basically in the middle of the peninsula, and the rivers didn't flow to the ocean. And so at some point starting 2 million years ago, more or less, that's what the geology people think, the rivers started transitioning from this system where you have lots of small streams going into a lake, into these open rivers that drain to the Atlantic Ocean or the Mediterranean Sea. And we think this provides plenty of opportunity for them to diverge in different lakes and then hybridize when the rivers got to this structure nowadays, yes. Yep.
Okay, I was wondering about the population that you, when you're doing the G-stops.
Yep.
So 2 individuals at first, and you said, okay, this is it, and then you sampled a whole bunch more individuals, and you said, okay, it's actually quite variable.
So you're talking about this.
Yeah.
So in this population we sampled 9. So what I showed when I didn't have many individuals is the 9 together, and then when you actually calculate it per individual, you see the individual variation.
So I was wondering if in your other populations, when you sample individuals on their own, if you see similar levels of individual level variation.
No, actually for this case in particular, if you calculate this per individual, they're all pretty similar. Yeah, and this is another thing that makes us think this is really ancient in the sense that they all seem to have pretty similar proportions in combination with the fact that they're not connected anymore. And I have to go now.
Hi, everyone. Thank you for being here today. My name is Katherine. I'm based at the University of Guelph. And I'm going to be talking to you a little bit about some comparisons of clustering algorithms we've done as part of a group effort. So as we all know who are here in this session, hybridization is pretty ubiquitous in nature. It happens in pretty much every major eukaryotic group, from animals to plants to fungi. And a lot of the time we start with looking at clusters in our data, so population structured data. So from here on out, I'm going to call these clusters K. And there's a few different ways to do this. So you can start with looking at a principal components analysis. You can look at maximum likelihood models, something like admixture. And then we have our Bayesian models, things like structure and entropy. And when I first started in this field, one of the things that I noticed was that in the literature, people often use these somewhat interchangeably, and they don't necessarily say why they're using structure versus admixture versus another program. So I was curious about, there's the chime, about what are the differences between these programs, and when is more appropriate than another, depending on what kind of data you have and what kind of questions you're asking. So I decided to look at 4 programs of interest. I'm well aware that there's lots more out there, but these are the 4 we decided to focus on because of the types of questions we ask in our lab. So I'm focusing on structure, fast structure, admixture, and entropy, which we will continue to see in this order. And before I started, I wanted to come into this sort of naively and sort of look at these programs for the very first time, run them for the very first time, without asking someone who had actually ever run them before as a naive user. So I looked in the literature and basically saw, okay, what are the differences between these programs that are reported? So there's a lot of similarities, but there are some differences. So all of these are Bayesian, except for admixture, which is a maximum likelihood model. All of them only accept hard genotype calls, except for entropy, which does accept likelihoods. And then we sort of have a gradient of things like the ability to detect weak structure, how computationally intensive the work is, the model specifications for the user in terms of what you can parameterize in your model. And then I've also just included here citations to date for each program based on their original paper. And it's important to note here, what I tested is not mixed-ploidy data sets, but it is worth noting that fast structure and admixture cannot analyze anything other than diploids, where structure and entropy do purport to take mixed-ploidy data sets. So what I'm going to show you here is there's tons and tons of results that we can pull from something like this. I'm going to show you a subset. But what I want to show is, so we tested all programs with the same data sets. So first we started with a bunch of simulated data sets that my colleague Erin made in SLIM, and we have just shy of 1,200 data sets with almost 300 of each sort of known clustering, so 2K, 3K, 4K, and 5K. These had a sort of varying number of SNPs. These were hard genotype calls. We simulated hybridization using a varying migration rate. And what I did was I tested K equaled the modeled K value up to 2. So in each case, for example, when we had 2K, I tested up to 4K, and so on. We also tested this across a real-world data set, which you might have heard Liz talk about a few moments ago. So this was a subset of the total data set that Liz was talking about. So in this case, I only pulled these leuciscid minnows from one watershed, where that watershed is reasonably isolated. Not isolated, but like you don't get a lot of movement between them. In this case, we had 38 individuals, just over 9,400 SNPs. We had genotype likelihoods. And in this case, we had a hypothesis of K equals 4 because there were 4 species identified morphologically. And in this case, we tested up to K equals 10 because, again, we know that there are tons of different species in this system. So some of the results I'm about to show you are in the form of a confusion matrix. What this is that on the x-axis we have the modeled K value, and on the y-axis we will have the K value predicted by the program, and the numbers you see will be the frequency of data sets that appear sort of in each of those sort of modeled by predictive boxes. Now, so for these methods, I'm going to show you some things, and I don't want you to panic, because there's lots of caveats here, so I don't want you to panic when you see these and you're like, oh my god, it doesn't work. No, it's fine. So first I'm going to show you structure. So, and again, as I said, I went into this purposefully as a naive user to test not only the sort of biological results that you get from these, but the actual feasibility as a researcher to use the programs. Anyone who's used structure before can tell you it takes forever. So on purpose, I had like a sort of a time limit of 14 days. And of our 1,188 data sets, only 110 runs actually finished. We didn't even get any of our replicates finished. And these are on data sets with very relatively small numbers of SNPs too, so this was quite something. Now to estimate K, structure says to use ln probabilities to then calculate your posterior probabilities. But what we found actually was with these data sets, that can actually lead to massive underflow in the computation environment. So what I mean by that is that the numbers were actually too small for any of the programs to calculate. So what this meant was I actually got very few results from those 110 runs because the computer literally could not calculate them. So again, I don't want you to panic by seeing how far outside of these hopeful yellow boxes these results are, because the Ivano method exists. And again, if you've used structure before, you've probably come across the Ivano method. This is where instead of using those posterior probabilities, you use something called delta K. And delta K is the mean K likelihood times the second order rate of change divided by the standard deviation of the K likelihood. Now, because I don't have replicates, I can't actually run the calculations in this instance for Ivano, but what we can do is we can look at the distributions of the ln probabilities, which is what the Ivano group used as their justification for coming up with that equation. What that looks like is that, so from left to right here, I have, again, my data sets. These are the results of the data sets that finished, split by the sort of modeled K value. And what you're looking for, and what if the Ivano group, I don't know if you can see, you can't see the mouse, but what you're looking for is that basically a large shift in that distribution at the supposed K value. So you can see that like in that first plot on the left there where we have the most replicates to look at, there is a shift in that distribution at K equals 2. That's what they're looking for. So again, I couldn't do the actual calculations on these data, but we do see that shift there, which leads me to think, okay, like that is what we would be seeing if we were able to have run all of those replicates. So next I'm going to show you what this looked like for fast structure. So fast structure, as its name implies, is very, very fast. So with a simple prior, all runs finish sequentially in about a week. This was what we got. So it overestimated in a lot of cases. Yeah, basically we find that it does hit our predicted, or our modeled K value a lot of the time, but the vast majority of the time it overestimates. And again, I only modeled K plus 2 in all cases, so it's entirely possible that these distributions could have gone higher. And again, after I show you the admixture results, we'll discuss a few caveats of this situation. So admixture is incredibly fast. A whole run of these data sets finished for the 2K in less than an hour. In each case, I did 10 replicates per data set, so what I'm showing you here is the summarized results across a given data set. No, don't panic, okay? Don't panic. This looks bad. So yeah, so it looks like, again, admixture is really overestimating the K value here. But one thing I noticed, so there's two caveats. One is that I noticed when I was looking at the cross-validation error distributions, which is what the admixture manual tells you to use, is that they actually look shockingly similar to the lawn probability distributions that structure outputs. And I thought, okay, can we take those same principles and apply the Ivano method to admixture? And when we do, we actually seem to get a sort of more reliable, or not necessarily more reliable, but more what our predictions would suggest, series of k-value estimations. So what I am going to say about this is that because our simulations didn't necessarily behave the way we expected, we are going to rerun these with some different simulations with some higher levels of SNPs, just to see, sort of why are we getting such high overestimations in these cases. So what I want to draw your attention to is basically how much variation there still is across these programs in terms of even if our simulations were overestimating or overmodeling our expected k values, we're still getting a lot of variation across programs in terms of their usability and what they're outputting, which is quite something. So we weren't actually able to do the simulations with entropy because the way we made the simulations is they only had hard calls. Entropy does technically take hard calls, but the input file format really doesn't like that. So we were struggling to make it work. So that is going to be one of our next steps, is that we are going to simulate genotype likelihoods in order to use this. So now I'm going to show you the results we got for the real-world data set. So I'm going to show you a series of structure-style plots. So this is what we got for structure with the Minnow data set. So again, by the Ivano method, we got k equals 5. Makes sense. Clusters look nice. For fast structure, I'm showing you k equals 4 here, which popped out the most. But interestingly, fast structure is theoretically not supposed to need repeated iterations. It's supposed to do its own iterations when you run the program. But I thought, for kicks, I'll run it multiple times with the exact same parameters, and it spat out different results every time I ran it, which was a little weird. I did not expect that, which suggests to me that it might not be converging as well as one would hope. With admixture, again, by the admixture instructions, it indicated k equals 6. By the Ivano method, which I tried again, it indicated a very strong, very clear k equals 4, which was interesting. And then with entropy, it actually, by model DIC, it actually indicated k equals 6, and seems to be picking up on maybe a few more like subtleties or subtle structure in the data. So what I'm going to show you now is, instead of showing it by cluster, I'm showing it by individual. So along the x-axis here, you can look sort of vertically and see the same individual across all 4 programs. And what we see is that all 4 programs do give us a very similar picture of what these populations look like, or what these groups look like, with some programs maybe picking out some subtle structure sort of a little bit better than others. So to very quickly summarize sort of across, and again, this is like purely based on my experience using these programs. So we still have some more tests to run, but it seems like in general, fast structure and admixture might have sort of a little bit of a problem maybe with, especially as you get increasing complexity of your data sets. Feasibility, admixture is super easy. If anyone has done these before, admixture is super easy to run. Fast structure, if you can get it working, is super easy to run after that point. Structure and entropy are borderline user hostile, if you've never used any of these things before. Yeah. And then in terms of computation needs, again, fast structure and admixture are super fast, and structure and entropy take a very long time. So some general recommendations, I would say, and I mean, again, we're going to do a lot more tests to sort of verify all of these. If you need fast results, use admixture. If you need deep results, or if your data set has mixed-ploidy, you're going to want to use structure or entropy. If you have genotype likelihoods, you're going to want to use entropy because that's the only program of these 4, at least, that can handle it. You're going to want to parallelize or otherwise run your replicates simultaneously, if you're not already. And in general, you could start with admixture and then use entropy. So start to get your range of k values and then use something deeper if you want that deeper data. So very quickly, just some conclusions. Future work, as I said, we're going to be running a lot more tests. And in thinking about the future, our data sets keep getting bigger with higher numbers of SNPs, more individuals as these technologies become more accessible and cheaper. And I think we sort of need to think about, in terms of the computation needs, what this is going to look like in the future and what is realistic, what is accessible, especially as we have to realize that not every lab has access to high-powered computing all the time. So we have to sort of think about that. Like to acknowledge these absolutely fantastic folks, Digital Research Alliance of Canada and Food from Thought from the funding. And I would be very happy to take questions if there's time. Yeah.
In my experience, structure tends to scale its runtime with both number of SNPs and number of individuals. Have you considered like flipping it and trying lots of SNPs and less individuals?
Yeah, so that was actually one of the reasons why I decided to use the reduced minnow data set instead of the full one that Liz was talking about, because we could barely get a run of the full data set done in 28 days, which is our current limit for our computation access. So yeah, totally. I think that's definitely something worth doing is like, yeah, flipping the axes on individuals versus SNPs, for sure. Yeah.
Nice talk.
Thank you.
Incorporate isolation by distance to your simulations. Don't we know that can use some kind of this kind of softwares, and it would be nice because it's some hybrids on that kind of smooth gradation, so we can have a lot of isolation by distance.
Yeah, that's a great suggestion. Thank you. Yeah, and there's definitely, like I say, I didn't get into any of the comparisons with the differing migration rate that we simulated, but yeah, for sure, that's definitely something to consider because, as you say, it can totally influence your results. Yeah. I think I have time for one more. Sorry. Oh no, maybe I don't. Talk to me after. Thank you.
Okay. Hi everyone. My name is Pia. I'm currently a PhD student at Yale University. Today I'll be presenting my undergraduate honors thesis from the University at Buffalo, where I worked with Trevor Kravenhoft and Dan McGuigan. The title of my talk today is Mosaic Hybridization in a Trilineage Fish Hybrid Zone with a comparison of rat-seq and low-coverage whole genome sequencing. We are all aware in this hybridization session that speciation does not always lead to complete reproductive isolation, and that allows for interbreeding of species and the formation of hybrid zones. In freshwater systems, physical and ecological dispersal barriers created via river basin rearrangement over time can lead to fragmented species distributions. In river and streamfish populations, secondary contact and hybridization can arise when river courses shift. In this project, I've been working on a secondary contact hybrid zone between the Johnny darter Etheostoma nigrum and the tessellated darter Etheostoma olmstedii in a hybrid zone in western New York. Darters are freshwater fish clade of about 250 species that are distributed broadly across eastern North America. Etheostoma nigrum and olmstedii are two of the most widespread species, actually spanning most of this area in eastern North America. They do share an area of range overlap that is located in western New York. Previous morphology-based analyses of the contact zone that I've been working on showed that there's habitat partitioning in the area of range overlap, where Etheostoma nigrum tends to populate smaller river tributaries, like very narrow streams, and olmstedii is found mainly in the larger river main stem. There's additional morphological evidence from the 1960s that identified hybrid individuals based on morphology from this area that we've been working on. So this is a quick overview of our study site located in western New York, if you're familiar in the Rochester area. The two species that we've been working on diverged from a common ancestor about 3 to 5 million years ago, and they're broadly split into two drainage basins: the Atlantic coast with Etheostoma olmstedii that will be colored in orange throughout this talk, and Etheostoma nigrum in the Mississippi River basin colored in green throughout this talk. The two species share a fairly large area of range overlap in the Lake Ontario and St. Lawrence River basin. And this area of range overlap is certainly an area of secondary contact, as glaciers covered this area until about 13 to 20,000 years ago. Previous phylogenetic analyses revealed two deeply divergent Etheostoma nigrum lineages from within the contact zone that formed because Etheostoma nigrum followed two distinct dispersal routes following the retreat of glaciation. So therefore, Etheostoma nigrum and olmstedii form a very unusual trilineage secondary contact hybrid zone in western New York. Before I get into the results of my analysis, I want to give you a quick overview of my dataset design. For this project, I used 3 different datasets: a low-coverage whole genome sequence dataset collected in 2021, a DD-RAD dataset from 2017, as well as a mitochondrial DNA dataset from 2017 as well. There are 26 individuals that are shared between all 3 datasets. And as you've seen in the title, we took a shot at comparing sequencing approaches. So to do that, we applied identical bioinformatic pipelines to our low-coverage whole genome sequence dataset and our DD-RAD sequence dataset as a reduced representation method. So what are the main questions I'm trying to address here? First, is there broad-scale collapse or maintenance of species boundaries in our secondary contact hybrid zone? Do we find genetic evidence for the hypotheses of ecological niche partitioning by stream size? Third, what are the spatial patterns of hybridization? And lastly, how do low-coverage whole genome sequencing and RAD-seq compare for the inference of population structure, admixture, and genomic differentiation? So from our PCA analyses of the RAD-seq and the low-coverage data sets, we're able to see the broad patterns of population structure in our hybrid zone. There are two focal streams that I'd like to point out here. These two streams are from the heart of our hybrid zone. These are Oatka and Hania creeks in yellow and dark blue colors. These two streams have individuals that cluster on both ends of the PC1 axis, where, from your perspective on that right end, is Ertheostoma nigrum ancestry, and on the left end is Ertheostoma olmstedii ancestry. What you notice here is that there are some individuals that are sort of scattered out in the intermediate range of the PC1 axis. So this is our first hint that there's something going on with hybrids in these streams, actually. Before I continue with the nuclear DNA data sets, I'd like to take a quick step to look at our mitochondrial DNA data set, which provides even more evidence for active hybridization and actually the long-standing history of the secondary contact hybrid zone. We see distributed on the map pie charts that show the k=3 ancestry coefficients. The circle in the middle is the mitochondrial DNA haplotype, and the outside of that pie chart is the nuclear DNA ancestry. The main takeaway from this data set is that mitochondrial introgression is actually much more widespread in the secondary contact hybrid zone than nuclear introgression. This presence of widespread heterospecific mitochondrial DNA haplotypes, coupled with almost pure nuclear DNA ancestry in multiple streams, for example, here in the Upper Genesee River that is circled in yellow, we see that there is a long history of gene flow in the secondary contact hybrid zone. We hypothesize that this mitochondrial DNA introgression is a remnant of shifting hybrid zones that have stabilized over time with pure nuclear ancestry of, for example, here in the Upper Genesee nigrum ancestry, but the population still contains that Ertheostoma olmstedii mitochondrial haplotype. To further analyze the hybrid zone structure, we performed admixture analyses where k=3 was the best fit for the data. I will point out a few of the focal streams where we observe different flavors of hybridization with varying genomic outcomes. While dispersal between tributaries is limited, we also find that gene flow within a single stream can be highly restricted. In this example here, Bennett's Creek, we see that at upstream sites, so sites 2, 3, and 4, we have pure Ertheostoma nigrum ancestry, while at the furthest downstream site at site 1, we have pure Ertheostoma olmstedii ancestry. Between sites 1 and 2, there is a stretch of about 15 kilometers. The whole stream is just 20 kilometers in length. But in this 15-kilometer stretch, you notice we don't have any sampling sites. That's because we didn't find any Ertheostoma olmstedii or nigrum individuals in this stretch of Bennett's Creek. While we did find other darter species and other streamfish species, so this is, a bit striking here, at the same time, there's also no historical record of any olmstedii or nigrum in this stretch of the Bennett's Creek. The next flavor of this hybrid zone is found in Canisius Creek, where we observe a hybrid swarm with about 80% Ertheostoma olmstedii ancestry and 20% Ertheostoma nigrum ancestry that is consistent across all sampling sites. These admixture proportions are also stable across sampling years. To remind you, the DD-RAD dataset was collected in 2017, and the low-coverage dataset is from 2021. The last flavor of this hybrid zone is a pattern where Ertheostoma nigrum mainly populates upstream stretches of the streams and tributaries, while Ertheostoma olmstedii ancestry is mainly found in the larger and main stem streams of the system. We identified admixed individuals in proximity to confluences of smaller and larger streams in Oatka and Hania creeks, which, for example, in Oatka Creek, which is the lower two plots on the slide, you see that site 1 is where we found most admixture, and the difference between sites 1A and 1B is the sampling year. And you see that further upstream sites have basically pure Ertheostoma nigrum ancestry. So this pattern of hybridization repeats across the contact zone, forming this mosaic of hybridization sites, with hybridization happening mainly in proximity to stream confluences. So with these results, we can already answer 3 of the main questions that I asked earlier. First, we don't find that there's any broad scale of collapse of species boundaries, since we still, even in the streams where we have active hybridization, Ethiostoma olmstedii and nigrum maintain their sort of stream habitats where nigrum is only found at upstream sites where the stream width is very narrow and olmstedii is in a larger mainstream. So I already went on to the second result, but we can support the hypotheses of ecological niche partitioning by stream size with Ethiostoma nigrum in the tributaries and olmstedii in the larger main stem streams. Lastly, what are the spatial patterns of hybridization? We see that hybridization sites are always proximate to stream confluences, forming this mosaic of repeated hybridization across the contact zone. There's still one question left that we need to answer in this talk. Our comparison of sequencing methods. So from our results of the PCA and the admixture analyses, where I didn't go too much into detail with the comparison, we see that there are similar admixture proportions and PCA results for our 2 data sets. We find that the ancestry coefficients of our RADseq and low coverage data sets are highly correlated. However, individuals with more missing RADseq genotypes exhibit greater discrepancy between their RADseq and low coverage whole genome sequence ancestry coefficients. On the other hand, the 2 sequencing approaches actually differ substantially in their estimates of population differentiation between Ethiostoma olmstedii and Ethiostoma nigrum. We performed FST estimates for the 26 individuals that are shared between both data sets, and we also only analyzed FST for the same overlapping sliding windows. The Ethiostoma olmstedii and nigrum individuals that were included in these analyses were the non-admixed ones. What we can see from these 2 plots here is that the low coverage dataset identifies broad shifts in FST across chromosomes that are not recognized by the ddRAD dataset, which you can see in this sort of washed out bottom plot here. Unsurprisingly, the FST estimates from the RADseq and the low coverage datasets show only a very weak positive correlation. And what we also found is that the FST estimates for the RADseq data are on average higher than for the low coverage data set. With that, we're able to answer the last open question that we had here. So overall, we found similar patterns of population structure and admixture from our 2 data sets. However, the whole genome sequence data and the reduced representation data differ substantially in their estimates of population differentiation. From these results, we conclude that as the cost of DNA sequencing continues to decline and reference genomes become more widely available, low coverage whole genome sequencing will probably overtake RAD sequencing as the preferred approach for population genetic studies in non-model organisms. With that, I'd like to acknowledge a ton of people that helped with this project, my former lab members in the Krabbenhoft lab at UB, our helping hands with fieldwork, our funding and computing resources, and specifically the UB Biological Sciences undergraduate honors program and the director Jessica Poulin. Without this project and my whole entry into research would have not been possible. Thanks a lot for listening, and I'll take any questions. Yes.
You might have said this and I dismissed it, but what was your coverage on your double digest RAD sequencing?
I don't recall specifically, to be honest. Roughly 15x, maybe. Don't pin me down on that. With low coverage data, I'm very confident that was 1.9x on average across all individuals and all sites.
Yeah, I guess my sort of other comment about that, like, I totally believe what you're showing, but do you lose the ability to diagnose individuals as to whether they're F1s or back crosses or whatever? And I guess is there a cost to sort of losing that resolution about the individuals versus the population?
So I'm just going to repeat the question to make sure I'm understanding correctly. You mean if there is, to estimate, for example, heterozygosity and hybrid indexes from these data?
If you're wanting to know for an individual, like at a certain part of the genome, are you an F1 or a back cross? I assume with a little coverage that you lose the ability to do that. But to get that with the double digest RAD, you
Yeah.
the trade-offs there?
Yeah, so what we've been thinking about is that the main sort of restriction with the dd-RAD data set is probably that the coverage is not spread out across the chromosome, right? You have to these randomly distributed RAD loci, whereas even if the coverage is really low, it's more valuable to have coverage spread across the whole genome to specifically miss out, for example, on adaptive loci. So that's what we think is actually the main reason why we find different results for the FST between the two sequencing approaches, where with ancestry coefficients, it doesn't make that big of a difference.